Okay, 215, let's go back into this part. So cytogenetics is studying the genetics of the cell. Cyto, cell, genetics being genetics. And this is a karyotype. What they do is they take typically a white blood cell, they pull it out, and as it's going through mitosis, while the chromosomes are all stuck together, they freeze it, they chip it open, and they line them all across, and they call it karyotype. And we'll show that. But what they'll do is they'll take all of these and they'll line them up by size. So they take the longest ones all the way down to the shortest ones, and then they find the little sex chromosome, the loner, and they put it over by itself. And we'll show you those too. So if you get this little tiny mutated fragment that's all by itself, that would be the Y. The X is the longer one. The X has full of really important genes. The Y is pretty much worthless, other than a little handful of genes, like the gene that puts hair on our ears, very important as a man. And that one that makes us male, which is kind of important, S or Y gene. But the cytogenetics, like I said, typically they'll take a lymphocyte, they'll culture it, split it apart. And then when they look at these, they look at the different sections. You have the long arm and the short arm when you're talking about the chromosomes. So the short arm is called the petite, and so you use the letter P. Where's my mouse? There we go. So the short arm is the petite, like that. You can see it's clearly petite. And then the other one's Q. Nothing special to help you remember the Q. So when they're naming these different sections, they can actually color them, and they don't come colored like this out of your body. They actually have to color code them, so artificially stain them. So they have these stripes, and that helps them identify the different sections. So starting right here at the center, it'll go 1, 2, 3, 4, 5, all the way out. So these bands, these different colored bands, get numbered. And they tell you on the short or the long arm, like you'll have the P1 for the petite arm, and then you have the Q1 for the short arm, or for the long arm, sorry. I feel like I skipped over one, did I? There it is. This is the right one, right? Okay. When you talk about these sections, there are a couple of special terms, locus and allele. Locus is like location. So where is it located at? So if you look at the locus for testicular feminization, would this be the short or the long arm? This is the long arm, and it actually covers several genes. So if you pull over here, it goes all the way from 1 all the way down to about roughly 23. So there's a big section that's responsible for that. When you talk about the allele is one little segment. Using the recipe situation, an allele isn't the actual recipe for like cookies, it's the type of cookie. So allele is the type. So let's say that I pull out a section, and this section here, the gene is for chocolate chip cookies. Are all chocolate chip cookies the same chocolate? No. I can have a white chocolate, and I can have an allele for white chocolate. I can have an allele for dark chocolate. I can have an allele for milk chocolate. They're all cookies. The recipe is pretty much identical. It's just basically different shades of chip. So allele is just naming a type of gene. With the gene, still the same recipe, what type of gene is it? And then polymorphism we'll actually talk about later. It's if you have polymorphism, it means more than 2 of these locations are affected. Usually you get affected genes that are further away from the center. Where's my Lua? I'm going to keep going. I'll just walk through this. So more of the flaws are usually down here. Down or up here towards the center is usually more tight. It's more constricted. It doesn't have as many errors. Down here you get lots of problems. Like when you see Fragile X, that happens because it's fragile. All we have to fix is the furthest point away from the chromosome. So you're more likely to see things like that repetitive. And this next slide shows you alleles. So like I said, you have the same gene here, here, and here. But because they're different alleles, there's different flavors of the gene. So you get the same flower, the only difference was the color. You have an allele for the petals on here, still making petals with the red petals. The allele for the petal here, but they're making white. The allele for the petals here, but they're making yellow. So all it's doing is changing the flavors, if you want to think of it that way, of the gene. A couple other words you'll see over and over again. A genotype versus a phenotype. A genotype is the actual physical recipe. The phenotype is what you get as a product. They're not always the same. So a genotype is the actual recipe, all the letters, exactly how it's supposed to be. The phenotype is what you actually get as the final product. Genotype is genetics. Phenotype is what you see. So, for instance, if your mom has blue eyes and your dad has brown eyes, you may have brown eyes, but you actually carry the gene for blue eyes. So is it possible that your child could have blue eyes? Sure is. So you get two brown-eyed parents and you have a blue-eyed child. Is that possible? It is. Yep. That means that both parents had to be carrying a gene for the blue eyes. But their phenotype was what? Brown eyes. They physically had brown eyes. PH, physical. The physical presentation. Gene, genotype. And we'll talk about this with blood types too, but a person with type A blood could actually be AA. They could have a gene for A on one side of the chromosome, a gene for A on the other side of the chromosome, and it being A. But they could have a gene for A and another one for O, which means they have no little structures on the blood cell, which means that overall the type is A. So either way, their phenotype is what kind of blood do they have, A or O. They have A. But their genotype here is AA or AO. Okay, next terms, homozygous and heterozygous. Homozygous means the same. Hetero is different. So if I say you get two genes for cookies, if you're homozygous, they're both milk chocolate chip cookies. If you're heterozygous, one's for milk chocolate, one's for white chocolate, or one's for milk chocolate, one's for dark chocolate. So if my genes are O and O, what's my genotype? This is my genotype, right? What's my genotype? I'm type O. To be O, be a type O period, you have to be homozygous for O. You have to have two of the O's. If I give you an A and an O, what's your genotype? AO. What's your phenotype? A. You have an A and an O. They're not the same. They're heterozygous. And we'll talk about this with mutations again. Some people, they carry the bad gene, like in breast cancer. You may have one bad gene, and if you're lucky, are you homozygous or heterozygous? Hetero, because you might have a good gene and one bad gene, and this good gene will actually overpower the bad one. If you're homozygous for a flawed gene, is that good or bad? That's bad. That means you have no copies of a good gene. Both your copies are exactly the same, and they're both broken, which will be sickle cell, which we'll talk about in just a little bit. They're homozygous for that flawed gene. When you look at Mendelian genetics, so Gregor Mendel was a monk. He originally studied bees, but bees were too complex because they were animals. So he started looking at pea plants and dragons, snapdragons. Is that type of pea plant? Snapdragon's a flower, isn't it? I just remember pea plants and snapdragons. But when he originally looked at these different things, he found the plants were easy to analyze because he could start mixing different colors of plants and he could predict the outcome. With the bees, he had no clue what was going on. Because he didn't look inside the nucleus and he didn't look at all of the genetics inside of there. He just looked at the, what? Genotype or phenotype? Phenotype is all he could see. He didn't have the tools to look at the genes. And we call, when you follow based on their phenotype and try and predict what's inside, we call that Mendelian genetics. We can take these single gene disorders we're going to talk about and run them through this Mendelian genetics. And anybody know what this is called? This square? Punnett square. We can look at what's going on with mom, what's going on with dad. We can mix through and predict what your chances of having a child with this kind of disorder, or this kind of hair, or this kind of eyes, or whatever. It's called the Punnett square. When you look at this Mendelian genetics, there are two terms you have to know. They're called dominant and recessive. Which one do you typically see? The dominant. So the dominant versus recessive. If I get one gene that's dominant and one that's recessive, which one do I see? The dominant. How is the only way I see recessive? Two recessives, exactly. So where are they, hetero or homozygous? They're homozygous for it. What do you see if you're homozygous for a dominant trait? You see the dominant trait. Do you have any chance at anything else? You're homozygous. Yeah, you have exactly that gene. There's no way you're getting anything but that dominant trait. Okay, and then these are principles we're going to go through, so I'm going to skip through it. You need to be able to draw a Punnett square. It's not really hard. I have to do one quick. we haven't done sickle cell yet, but I'll use that as an example. So with sickle cell disease, you have this little, I'm going to draw, that's the bad one, because little S and big S is pretty much the same. How about I'll just put G for good and then little g for not good. So which one of these would be dominant, you know? Yep, so this is dominant. Usually when they put a capital, it's dominant. And then the little one is recessive. Let's say that here's mom. Mom was born with a good G and a crappy G. Dad's the same way. Dad was born with a good gene and a crappy gene. For you to have sickle cell anemia, you have to be recessive, and you also have to be homozygous. Do either of these people have sickle cell disease? Nope, because they're both what? They're both heterozygous, right? This is interesting about sickle cell disease. You actually have something called sickle cell trait, which means that you have a good gene and one bad gene. One bad gene. This is interesting because sickle cell trait means that you're immune to, what's that disease? Malaria. Yeah, so mom and dad, they're not going to get malaria. Good for them. The sucky thing is that if they have a child, what's the chance of their child having sickle cell disease? So you just pull this out. Mom gives a good gene, dad gives a good gene. Is that kid going to have sickle cell disease? Are they going to have sickle cell trait? Nope. This is a normal kid with neither. So, how about this one? Mom gives a bad gene, dad gives a good gene. What's that kid have? Yep, so this kid has the trait. How about this one down here? Dad gives a bad gene, mom gives a good one. What's it have? Has the trait also. So we've got these. Let me go like this. They have sickle cell trait. This one has nothing. This one gets a bad gene from dad, bad gene from mom. What's the problem? Yep, they have anemia. Sickle cell anemia. So what's the chances of having a child born with sickle cell anemia if you're both heterozygous? 25%. What's the chance of having a child born with sickle cell trait? 50%. And then what about a normal child? 25%.
The 50% is that because the small gene, well, I guess the bad gene also carries some sort of other protein, so it has both that negative
For the trait?
right. So the negative sort of aspect in that it's a carrier at the same time it's able to create a protein that helps the area.
Yeah, its protein for hemoglobin is normal enough, but there's just some little characteristics that malaria doesn't like the environment. It's okay because it still binds to oxygen, it doesn't change shape, but there's just something about it that malaria doesn't respond to. Yep, so when they have sickle cell trait, they still make normal hemoglobin, like everybody else. But there's something about that hemoglobin chain that's changed just enough that the malaria parasite doesn't like eat it. It's repelled by this stuff, so it doesn't want to hang out there. What's the other word for these people? Can their kids get it?
Yeah.
They're carriers, exactly. So they're carriers. Is this kid a carrier? Nope. Is this kid carrying it? Yes. So what's the chances of having a child that carries that trait on? 75% are going to be carriers. One will actually have the full-blown disease, two have the trait, and won't even be a carrier at all. Right, oops, I forgot that was on the next slide, so there it is. So, sickle cell disease, it's a single gene disorder. What do you know about the kid that has it? They're homozygous recessive. Exactly. It's a homozygous recessive disease. I'm going to put the slide in here. And this is what the cells will actually look like. So the parasite gets in, you get bitten by a mosquito. The parasite, part of its life cycle is actually in the saliva of the mosquito, but it's kind of like in this dormant stage. Then when it gets injected into this nice warm body, it gets circulated in the body, goes to the liver and says, ooh, this is great. We got all that sugar, we got all the fat, we got all the nutrients here. And so it's growing, developing, gets in the blood, hangs out in the blood cells, And then when it fills with the blood cells, it pops. It destroys the blood cells. That's how malaria works. But something about, they have sickle cell trait, it goes to get inside the red blood cell and it can't get in there. It's uncomfortable in there. So what's going to take care of these things? They're just flowing through the blood, susceptible to attack by what part of the body? The immune system. The immune system can destroy it. But if they get inside a red blood cell, you can see that they start growing and they explode the cell. Sickle cell trait prevents that. Okay, so some terms we're going to play with again. This is still kind of terminology, but patterns of inheritance. Single gene. Single genes are easy because you can take that Punnett square and figure them out. The ones that are tough are these polygenetic ones. So what's that mean? Polygenetic. Yep, many genes. So it's an added effect. They have lots of genes that are playing in here. If you're lucky, sometimes you have a good gene on a totally different chromosome that hides the characteristics of the flawed genes. And when we talk about cancer, we'll talk about this too. You may have a flawed gene that makes you susceptible to cancer, but you may have a really strong gene somewhere else that hides those characteristics. So multifactorial means multiple alleles, different loci. So it's not just the different genes, it's also the alleles that play into it. Epistasis, like I was talking about, you may have one really good gene that actually hides the effects of some other crappy gene in your genetics. When we talk about multiple alleles, we'll actually talk about different genes that can work together too, like the ABO blood type. You can have a type A gene, you can have a type B gene. Which one's dominant? They're both equally dominant. So what type of phenotype do you get from having an A gene and a B gene? AB, yeah? We call it codominant. You can have complementary genes where they depend on each other. They're like table legs. If you have a table that's built for 4 legs and you have one bad one, what's going to happen to the table? Even though the other 3 legs are good, what's going to happen? It's wobbly and could topple over. So complementary genes means they all have to work together for any of them to work properly. Kind of like almost like a synergistic effect. And then collaborative genes are when 2 genes affect or work together and have the same outcome. We're not going to do a lot of pedigree, but there are a couple things you want to remember when you're looking at pedigrees. Pedigrees are these long chains where you look at the original family and then branching off. And typically you refer to the family tree. So you have these original branches. When you look at the shapes on here, you want to know the difference between male and female. I always remember that the square ones are what? Male. Because I was told that men are always blockheads, right? So there's a square. I feel like I can get away with ripping on men, but if I rip on anybody female, man, I would be out for it. So I make a lot of male jokes like the blockhead things or wolfing ducks and dogs. Do you remember that? I can go with that. Did I tell you my brain story about women and men in a brain store? Oh. Man and woman walk into a brain store. I got to tell it. So, man and woman walk into a brain store, and they're looking for a brand new brain. The clerk says, okay, the male brains are over here on this wall, and they start at $500,000 and they go up. The women's brains are over on this side. They start at $5 and they go up. And the man looks at his wife and goes, uh-huh. And the clerk says, oh, don't get me wrong. It's because the male brains haven't been used. You thought it was going to turn it around. Wrong. Anyway, so you always remember the male or blockheads, and when you see a square, it's the male. And of course, the round is the female. Usually when you see one's shaded in or darkened, that means that they had the disorder. When you see a slash through it, guess what happens then? They die. The proband is the first person in the family that had the disease. Typically, or the first person in record that has the disease. And if you followed it back, way, way, way back as far as you possibly could, the proband started like partway through this huge family tree, they probably started as a spontaneous mutation. But the proband is going to pass down generation to generation. When you look, you have a branch that comes out. Each one of these branches down here are a child. What happened there? This is not a direct child, it's a in-law, right? So this one, the red block, got married to this woman. So how many kids does this family have? How many biological children do they have? Yep, so they had 5 females and 1 male. 2 males. 1, 2, 3, 4 females, 1, 2 males. So 6 kids. And so how many of the kids, what percent of the kids have the disease? 50%. 80%. I have friends in Illinois, and I always pick on people when we do this, but that store, WALMART, I pronounce that. Walmart, right. And she said Walmart. I don't know how many times I said, what? She said Walmart. Walmart? And my wife figured this out, so when I have somebody repeat, she's like, shut up. Anyway, so, and then you can see, with this one, pass it down. How many kids does this couple have? So they had 1, 2, 3, 4, 5, 6. What percent got it? 7. Sorry, my arm's in the way. I'm going this way, and my arm stays that last one. So 7. About what percent got it?
7.
3 sevens, roughly 50%. Roughly 50%. So you can see that in this family, this disease, if you're going to have a child, what are the chances of your child getting it? About 50%. Did it matter what the sex was? Nope. In this generation, you can see 2 girls, 1 boy. The next generation, 2 boys, 1 girl. It didn't matter what the sex was. So you can follow it down generation, generation, generation. You can see whether you're going to have a boy or a girl, you have a what percent chance of having this disease? 50%. And that's Tay-Sachs would actually do this pedigree. So it doesn't matter if it's male or female, you still have a 50% chance. Okay. Some of the different types we'll talk about. If you look at a pedigree, we're going to talk about Huntington's later, so you don't have to write all the details down. But Huntington's, when you look at them, here you have who? Male and female. Who got the disease? The female. And if you look, what percent of the kids get the disease? About 50% of the kids. Yep. Huntington's is like that. About 50% of the children can get it. It's more common to have males, but it's not because it's a male-specific disease, because females can get it too. If you watch House, it's females, right? So you look at the progression. What's really bad about some of these, like autosomal dominant Huntington's, is that each generation it gets worse than the one before. And when we talk about it, you'll find that they're up in their 30s, like 30s and 40s before they actually have symptoms. What have they probably done by then? Had kids. So if they got it, then there's a 50% chance their kid got it too. If they didn't get it, then their kid's pretty much in the clear. So Huntington's disease. All right, now we're going to go into specific diseases. So the first group are called single gene disorders. And it's rare that I put always anywhere, but I kind of stressed it here. So single gene disorders always follow Mendelian genetics. They're always going to follow Mendelian genetics. So they call them Mendelian disorders. You can usually follow them through a pedigree too. So if you can follow the generations down and see who got it, who didn't get it. So as we talk about this, we'll talk about different types. We'll talk about autosomal dominant. We'll talk about autosomal recessive. When I say autosomes, I'm talking about, of your 46 chromosomes, I'm talking about the first 22 pairs. So 46 chromosomes.
We have a question on the Huntington side. So you said each generation gets worse than the one before?
Yep.
But if the chance is 50%, each time how, the symptoms are worse?
The symptoms get worse.
Oh.
Yep, the symptoms get worse, right. So every generation, the symptoms get worse than the generation before. Yep. Okay. So we'll talk about the autosomal link, which are the 46 chromosomes. Remember, we have 46 pairs. So 23 here, 23 here. The first 22 of each side are called autosomes. What's the last one's called? Sex chromosomes. So we'll talk about how this goes through autosomes and also how they're associated with sex. Typically when it's sex linked, it's because of the X chromosome. Who's part of it? Doesn't matter. So Exactly. Do men have X's? Yep. So men have X's too. If the baby's born male, who determined that? The male, yeah. But yeah, the man carries the X too, so he could pass that problem down. The nice thing about being female is that you have 2 copies of this X, which means if this one's broken, you got a backup copy, right? With a man, it's X and Y, so if the X is broken, you've got nothing. You've got 3. You can blame Dad for that. Right. When you look at these single genes, all of these are going to fall into abnormal proteins, or, so abnormal protein. It's going to be abnormal structural or abnormal enzyme. All about protein, but abnormal structure or enzyme. So either a building block for the body or an enzyme to speed up reactions. And here are the first ones. So autosomal dominant disorders. So a good example of this is collagen. Would that be an enzyme or a structure? It's a structure, right. Collagen is a building block. It helps you form bones and skin and everything else. So autosomal dominance. I'm so excited. Collagen, you got it right. Yay. Okay, so autosomal dominant. And whoops, another key to write down is the age of onset varies. So you can be older when you start seeing symptoms. When we talk about autosomal recessive, these are the things that are going to poke out. With autosomal recessive, usually you see it right from birth. If autosomal dominant is talking typically about structures, take a guess what autosomal recessive is going to rotate around. Enzymes. Yeah. So these usually revolve around structures, things like collagen, building blocks. Huntington's will actually fall into this. Huntington's affect proteins in the brain, structural proteins. You start building up plaques and it causes problems. Osteogenesis imperfecta is another one. Let's talk about bone imperfections. Do you think enzymes or structures when you think of bones? Structures. It's structural is what you really want to keep in mind. So when you see dominant, think structure down here. I should have probably bolded it and underlined it, but I didn't. I can't fit my page. When we look at something like this, we look at its recurrence risk. So what are the odds of a parent having it and then the kid having it? And I already told you one of them was Huntington's. What would be the odds of the kid having it? 50%. Yeah. So odds of getting it are 50%. When you look at the recurrence risk, this is what screws a lot of people up, isn't it? If a couple has a kid and the kid has a disease, and it's a 50% chance, what's the chance of the second kid having the disease? It's still 50%. Each kid gets that 50% chance. So they might have 4 kids in a row that none of them have the disease. When the 5th kid comes along, what are the chances of the 5th kid getting it? Still 50%. It's still 50% every time. And that's the recurrence. What are the odds of it happening? Recurrence risk. And the recurrence risk of a dominant, autosomal dominant disease is 50%. Here's another example, achondroplasia. This is a fancy word for what? Dwarfism. Yeah. Word for dwarfism. I don't know what happened to that picture on that slide. Everything's like in a haze, but anyway, it's the most common form of dwarfism, achondroplasia, and autosomal dominant, which means if the parents, let's say that dad is a dwarf, mom's full height, what's the chance of the kid getting it? 50%. Yeah, 50% chance. What's interesting, and this is a hard one for some people to wrap their mind around too, is that there have been enough flaws. I'm not going to make you remember. What would this mean, by the way? If it's localized to the 4p16. If the petite part comes to number 4, what's that? The L word. It's this locus. The 4th chromosome, the short limb, is the 16th locus. So we've identified where this defect is. So, anyway, what people have a rough time wrapping their mind around is that if two dwarfs, they both carry this gene, right? They have one bad gene for which gave them the characteristics. If they have children, if the child gets both bad genes from the parents, it's usually not compatible with life. And a lot of people have a problem understanding that. Like, well, two dwarfs can have children. Yeah, they can. But if they both got the defective gene, then it's not compatible with life. Which means that one parent gave the bad gene and the other parent had to give the unflawed gene for the child to actually survive. Did somebody have a question? I saw a hand. Discussion. They don't really know. They just know it is. Yeah, it's still kind of mysterious. I think that's on another slide that I have coming up later too. But yeah, it was just one of those interesting things. And which leads me to the next thing. Reproduction for our human species is just like a miracle. It's amazing because back when I was learning all this stuff, it was roughly 1 in 3 conceptions actually led to a birth. So if a woman had a child, odds are that she could have been pregnant 2 times before and not even known it. Could have, been fertilized twice before. And then that third one. And I was listening to Dr. Radio a couple weeks ago, and they said it's more like less than 20%. So roughly 1 in 5 conceptions actually leads to a birth. And they think this because of all the stuff we didn't know. She could go a month into development of the child and something could be wrong with it and it actually spontaneously aborts and she wouldn't have known it. She would have just lost it in a regular menstrual cycle. So it's kind of interesting in looking at how flawed our genetics are and how hard it is actually for us to conceive. And then if you want to throw all the stuff about estrogens in the water and all that in there, it makes it even worse. But so when you look at achondroplasia passed down, is it recessive or dominant disorder? It's dominant. So how many genes, flawed genes, do they have to get? Just 1. So if they get 1 flawed gene from a parent, then they get the disorder. So the genotype is the 1 bad, 1 good. The phenotype is always going to be that they have short stature or delivery. If they get 2 genes, the 2 bad genes, then it's lethal. So altogether, if you look at the whole overall perspective, 25% survival rate for an embryo of 2 parents that have the genes.
Where did the last slide right there said 80% are spontaneous?
On the last slide? Yeah, there can be another spontaneous mutation. That means that 2 adult that are full-size adults, they could actually have a dwarf because of the spontaneous mutation too. So flaws happen.
80% of dwarfs aren't carriers yet?
They're new mutations. Yeah, there's a lot of new mutations. And part of that reason could be because if dwarf parents get together, what's most likely going to happen? Yeah, the embryo's not going to make it. And we're going to talk about some of the flaws later about why embryos don't make it too. Here's another one. So this is called polydactyly. Extra fingers, great piano player. I can pick on this because I'm, what am I, the 4th generation out of the Amish community. Yeah, I look Amish, right? It's the beard. I hate growing facial hair, so you'll never see that one. But yeah, it's kind of interesting, and this is just like Tay-Saxon Oshkosh's Jew population. We get a lot of polydactyly. So too many fingers, also too many toes. And this next concept called expression. So it's how much does it show? This person could have 6 fingers and 6 toes on every leg. They might have just 1 extra leg. So in this situation, they got 2 feet. So just because you get the bad gene, you may have a little of the symptom, but you may not have full blown. I mean, even in dwarfism, there are different heights to dwarfism. It doesn't say you're going to be exactly 1 height. In case of expressivity, of course, polydactyly is one of them. And then another one is called von Recklinghausen disease. I think it's on the next slide. Yep, sure is. I'm taking a world religion class right now, and I sit at the back of the class, and it's weird because 2 of the students were my former students, and they like sit near me. And the first day, one kept looking over her shoulder, and she finally goes, "Why are you here?" Kicking the class. What? Why? because I'm human too. But, no, I didn't say that to her. I said, did you fail my class? She goes, no. I said, we're cool then. But von Recklinghausen disease, and the reason this came up is because someone was talking about this picture. I didn't say it in class, that would be that guy. But, oh, you're von Recklinghausen. He was talking about, there's a guy that they call him the Elephant Man, and he's alive, not the Elephant Man, but an Elephant Man in some part of Japan. When people give him money because it's good luck. When he was China, it was good luck. We thought that was kind of a blessing. And I thought, I don't want to say what it was, but it's called von Recklinghausen. And technically it's called neurofibromatosis 1. So the interesting thing about medicine is that a lot of times you get the name of the disease and then you get like the name of the doctor. And they're called eponyms. Like Cushing's disease or Addison's disease. So unfortunately, usually you have to remember both names. So, neurofibromatosis, and just the name itself tells you what's going on. What are these things? Extensions of neurons, right? And they're fibrous, very fibrous, so they have fibers that are extending out, and they actually end up looking like well. They can be really mild little bumps that expose these things called cafe au lait spots. Coffee with milk spots. So it looks like coffee spills. They can be a little bit more extreme. You can see where all these nerve endings are coming out, and they're all branching out. And then it can be extreme. And then someone asked, well, what, in one other class, why don't you just get plastic surgery? These are neurons. If you cut them, what could happen? There was sensation to that part of the body. It could cause chronic pain in that part of the body. So you have to decide, do you want to look like that, or do you want to potentially have chronic pain in your face that you don't even want to move for the rest of your life? So it's kind of a touchy subject. And they even know where it's at. But you need to remember autosomal dominant. They know that it's on the long arm chromosome 17. So what letter will be after this? And its expressivity varies a lot. From having the little dark spots, the cafe au lait spots, to having huge bumps extending out. Or it can actually cause tumors in the brain. And then I already mentioned Huntington's. So the one other thing I really want you to jot down about Huntington's is that what did I tell you happens from one generation to the next? It's worse. And the reason it's worse is because those three little codes, you have a CCG. Every generation adds more CCG codes. So it gets longer and longer and longer. And this rod keeps getting bigger and bigger. So symptoms get worse and worse. What are the odds of a kid having it to someone that has it? 50% chance. All they have to do is get that one bad gene from mom or dad and they get it. How about what age does it usually affect them? In their 30s to early 40s, yeah. Because it's called a CCG repeat. Those three little codon letters. It's like having this word here, the dog ran, or a sentence, the dog ran for one day, and then suddenly you start studying a dog. And it's the next generation is the dog ran for one day. And the next one is the dog, and then the dog. The longer that is, the worse the symptoms get. It's like it's making excessive plaques, and every generation makes more plaques than the one before, because that dog word, that codon. And their life expectancy is typically 20 years after they're diagnosed. So after the first symptoms start showing up, then they've got roughly 20 years. Anybody know what other diseases, to me, this looks like? What are the symptoms? What do they get? And you've probably seen it. In fact, there's a disease that I know you're familiar with where they get jerky movements. It looks a lot like Parkinson's. Yeah. And we'll talk about this in the brain. We'll compare Parkinson's to Huntington's. But they used to call it Huntington's chorea, which means it looks like they had these choreographed dance movements. Because they have these uncontrollable movements. So it looks like Parkinson's, but what's interesting about it is that they have cognitive symptoms like Alzheimer's. Memory starts going, mood starts changing. So it's like a blend between Parkinson's and Alzheimer's. And we'll talk about it again when we get into the brain. Some of the diseases this semester you'll see repeats. You have to know it for the genetic part. Oops. You have to know it for like the brain area. Okay. And then here's an interesting one, progeria. So sometimes you get these mutations. What was the other one I said that you could get spontaneous reaction you brought up? You get a spontaneous mutation that causes it. It wasn't because of the parents necessarily. Achondroplasia. Progeria is another one. Progeria is typically passed down from the dad. And it's not because dad had progeria, because these are not old people. They're children. I think they're both, they're both about 10 years of age. Have they had kids yet? No, unlikely not. This typically happens as a flaw to the sperm. Old sperm. So, just like when we talked about the female egg, it's been sitting in her body dormant for potentially 35 years. What disorder do we associate with the offspring of a 35-year-old or older woman? Down syndrome. And we don't point much at the sperm, but the sperm, the original germ cell, the sperm, has been reproducing for 35 or 40 years. Is it going to pick up flaws? Yeah. One of the flaws can actually lead to a mutation called progeria. All it takes is one bad gene in this situation. You get one bad gene from dad, and you've got progeria. And their body ages just like they're 60s or 70s. They just grow really fast. So they look old. They get a lot of old symptoms, like stiff joints, hip dislocation, heart disease. Basically, they're 10 years old and they're living like they're 60 or 70. Apparently, they dress like it. Reminds me of Mr. Rogers every time I see it. Okay, so we refer to it as genetically lethal because basically they're pre-programmed to die early. And their symptoms typically happen right around 2 years of age. They start seeing this progressive aging. I think this might be one of the last ones, Marfan's. So Marfan's syndrome, here collected, they're all structural. These guys are really, really tall. Doesn't have to be a guy. They have really long fingers, which they call arachnodactyly. Dactyly means finger. What do you think arachno? Spider-like fingers. Yeah, the fingers are real long. There's like spider legs there. I think that's in there somewhere. Yeah, arachnodactyly. So there's a fiber misshapen. You can refer to the scaffold protein. It holds everything together. It doesn't work properly, so they get joint problems. They get long fingers. Their legs get really long. Their arms get really long. Their eyes, they get cataracts really easy because the fiber that holds things together has a tendency to cause cataracts. And then the worst one, so the joints are not lethal. The long fingers aren't lethal. The eyes aren't lethal, but the heart is. So the same fibers that help hold the valves together and hold the wall together are weak. So they usually die by the time they're 30 or 40. And they think one of our presidents may have had this. Now, Lincoln. So if he had been assassinated, who knows? He could have died of a heart attack. I don't remember how old he was when he was assassinated, but I don't think he was barely into his 40s, was he? Okay. And didn't I already talk about recurrence risk? No, I felt like it did. So under recurrence risk, there's a special word here, consanguinity. Yeah, we're some kissing cousins from Kentucky. But consanguinity, that's when two people that are closely related mate. Because if you have a disorder in that family, presto, you automatically have that bad gene in both people. So it increases the risk of having that thing happen. Consanguinity. And in India, there are actually certain areas that there's so much inbreeding that they get flaws with basically the face structure. So microencephaly is what that's called. What does that mean? You see the brain compartment is small compared to the rest of the face. Small or underdeveloped cranial regions. Consanguinity. Next one's autosomal recessives. So everything below or before fell into the dominant category. Most of them was passed down from generation to generation, but in the case of progeria, what happened to them? This isn't your next one? I skipped the slide? Oh, the order that it printed out?
Yeah.
Okay.
Found it.
Okay, great. Oop. So autosomal recessive. Before all those were autosomal dominant, they all had to do with structural proteins. So you saw height issues, everything from Marfans where they're over tall to achondroplasia where they're really short. We saw characteristics like progeria where they age really quick. Structural proteins aren't very strong. Now with autosomal recessive, this is going to be about enzymes. They're not going to be able to process things very well. And like I said before, when you look at this, these are more early in life, where autosomal dominant can happen any stage of life. Typically with single gene autosomal recessive, you see it early in life, like Tay-Sachs. So some examples, color blindness, you see it early in life. Tay-Sachs was one I already gave you. What was another one I gave you that had to do with red blood cells? Sickle cell. Someone mentioned one that they test for at birth where they take a little blood sample, PKU, phenylketonuria, which is in a couple slides from now. But these are all enzymes. PKU, you can't process phenylalanine very well. You can't process it at all. It accumulates in your brain and it causes retardation. Sickle cell anemia, you can't form hemoglobin properly. Oh, and Tay-Sachs, that was the other one. What was it they couldn't process? Proteins, carbs, or lipids? Lipids, exactly. Okay. So with single gene disorders, when you look, typically recurrence risk is about 25%. So it's a little more rare. Why? Because the kid has to have two bad genes, has to have one from mom and from dad. So if you have dad as big D and little d, does dad have a disease? No. Dad had to have what? Two little d's to get it. How about mom? Big D and little d. Nope, she doesn't have it. But when you look at this Hunt square, what's this one going to be? Will they be a carrier? Nope. How about these two? What are they? They're carriers. They have the disease? Nope. Just this one that has a two little d's has the disease. And what is it? An enzyme or structure typically? It's an enzyme. Okay. So one of two types of enzymes, either catabolic or anabolic. What does catabolic mean? Building or breaking? Breaking. So in catabolic enzyme deficiencies like phenylketonuria, PKU, they can't process pieces of protein, amino acids, phenylalanine. They can't break it down, so this stuff accumulates in their brain, causing retardation. When you look at something like this, cell parts, Tay-Sachs would be one of these diseases. They call them lysosome storage diseases because that lipid gets in there. They can't break it down, so they can't do anything with it. It goes into the stomach of the cell and just fills it up. It's like you plugging in your stomach and just keep eating and eating and eating. If you can't push it down, what's going to happen? Well, you might be able to vomit, but otherwise it would rip your stomach and it would kill you. That's what happens to the cell. It gets so full of lipids, it's dying. And then anabolic, what do you associate that with? Breaking down or building up? Building up. Anabolic deficiency means that what? It's not that they're... So, this means exactly they can't make something. Cystic fibrosis, they can't make a special transporter for chloride. That slides in here in a little bit, but they can't make a special transporter. It's a little enzyme that moves chloride back and forth. If they can't make it, they can't transport chloride in and out. And we know from the fluid and electrolytes, where's chloride more abundant at, inside or outside the cell? Outside. What's it want to pull with it? What's it want to keep near it? Sodium. So if they can't pull sodium into the cell, it stays on the outside. And if it's on the skin, they taste salty. Or they actually can send electrical current through that and detect it. So those are just two extreme examples, catabolic versus anabolic. Penetrance. Penetrance is a lot like expressivity, but it's not exactly the same. So penetrance says that the number of children that should have it that actually get it. When you're talking about recurrence risk, you're talking about 25% of the kids are going to have this genotype for this disorder. Penetrance, sometimes the 25% the kid actually gets the bad gene, but they don't get the full-blown disorder. And this is the one we're going to talk about. It's called retinoblastoma. When you see OMA, what's it always tell you? It's a tumor, right? So you guess where it typically happens? In the retina. Yeah, so it happens in the retina. This is interesting. They call it incomplete penetrance because sometimes the kids get the gene for this tumor and they don't get the tumor. They're destined to have it. They have the gene for it. Something doesn't happen. They don't get it. It looks like a normal child. They don't get the tumors. It's called incomplete penetrance. It's kind of like once in a while you get lucky and hit the lottery. So it's a number of people that have the genotype that actually present with it. They don't express. Yeah, they don't. And I'm hesitant to use the word expressive, but you're right. They never show it. It's like people that would have 2 bad breast cancer genes, but they never get breast cancer. They lucked out. Which is the same example. Retinoblastoma, we're talking about tumors. Just like with cancer. It's called penetrance. Okay, so single gene disorders. The last group are called sex-linked disorders. And the sex-link can happen actually to the Y or the X. But the Y is so worthless, basically. Y-linked disorders, the worst one is the whole hair growing on the ears bit. So, not fatal. It's called hypertrichnosis penia. It's just excessive, and you don't have to know that because it's a worthless piece of information. It's just hairy earlobes, big deal. It's cool if you want to be a wolf for Halloween, like a wolf man, but other than that, not a lot of fun. But yeah, the Y chromosome is so worthless. It makes us male, and that's really the only good thing for us. And then, specifically between the sex-linked to the X. And can you see sex-linked disorders in women? Yeah. So when we talk about some of these, you have to think. But in a woman, what do you know about the X's? 2 are flawed. Both of them have to be flawed, typically, right? So with a man, all it takes is 1 flawed X, and he's got the disease. For a woman, she has to get both of them. So in a woman's situation, would this fall under a type of recessive or dominant? Recessive, because she had to have 2 bad genes, right? With a male, I'm hesitant to use the word dominant, but with a male, if it happens to the X chromosome, he's got it. There's no getting around it. Here's a trick. Can the dad pass down an X-linked disorder to his kid? Only if it's a girl. Exactly. Only if it's a girl. Because if dad has this X-linked disorder and he passes down the Y, was there anything wrong with the Y? So does the boy get it? Nope. But if he passes down the X, the girl will automatically be a what? Carrier. How could she have the disease? If mom gave it to her too. Exactly. So dad always passes it down, but you never see the X-linked disorder from a father to a son. Yep. So if dad has an X-linked disorder, he never passes it on to his son. So when you see the link followed through here, and then in this situation, did dad have it? Nope. So who passed it on? Mom. Mom passes it on. The son gets it automatically. What had to happen here? How'd that girl get it? Dad was a carrier. Yeah, you're right. What am I? Yeah. This is the right picture from the book. But you're right. Because if it's on the X, then what happened? He should have it. You know, I've been using this slide for 3 years. You're the first person pointed out. Yeah, it's right from the book for sex-linked disorders. Yep, there's a flaw there.
was checking out the they were trying to show that as a carrier state, but then they could see the different red boxes being.
Yep.
What about
Interesting. The arrow saying that just because this person doesn't have it, that person still can get it. So it can be passed down without even knowing. She's a carrier, right? So I guess don't pay attention to the pedigree, just look it over on the side. Bad history. So most X-linked disorders are recessive. And I already told you this, the male can't send it on to their son. But the sons of the female have a 50% recurrence risk. And then one good example is hemophilia. So when hemophilia is passed down, if mom doesn't have hemophilia, and she just has one gene for it, she passes it down. If she passes it to a girl, does the kid have it? Does the kid have the disease? Nope. His dad didn't do it, but what's this kid? Carrier. She doesn't have the disease. What about this boy? He gets a good X from mom, a good Y from dad? Nothing. What about the boy? Yep. So the boy is going to have it automatically. 25% chance. Huh? Hemophilia? There are 16 types of hemophilia. So I don't know if in general it's more common in Asian. And the next one is polygenetic and multifactorial. So polygenetic means it's many genes and many factors going into it too. This is a big tricky word that you have to read. Polygenetic means many genes. It's not one specific gene on one chromosome. So it's harder to predict if the kid's going to get it. It's multi-genes. Multifactorial means that it's genetics and environment have a play. And sometimes on the ultrasound you can actually see the genetics of it. You can see the flaws right in front, so they know ahead of time. It can be different degrees too. So you can have something like in the top where it's just one side, so it's unilateral. You can have it where it goes all the way up to the septum and penetrates into the nose. You can have, and here you can actually see all the way up into the nose. Or the whole roof of the mouth is completely gone. So it can be mild to severe. And it's a lot of different genes, like I said, it's not easily predicted. Okay. And then, I was going to skip. Oh, no. So, characteristics. Polygenetic members many genes. Multifactorial means it's not just the genetic driver like this. When we talk about things like this, we can count. Quantitative means nothing. The qualitative means the degree of it. The quantitative, you can actually count that up. So the different things are how severe it is, from mild to most severe. And I'm not going to make you plug in this, so the whole multifactorial inheritance chart, you don't even have to worry about that at all. It's just saying that based on the number of traits, the more this happens in a family, the more frequently it's going to happen, the more severe the symptoms are. The last section we're going through is chromosome abnormalities. So chromosome, you're looking at the whole chromosome. It's not one specific gene, it's not one specific like nucleotide pair or base pair. We're talking about a whole chip or a piece of a chromosome that's broken off, fragmented, falls apart, whatever happens. Right? And then the keys with the chromosomal leading or cause of mental retardation and miscarriage. And even with Down syndrome, we're going to talk about it being a chromosomal abnormality. Very few Down syndrome conceptions actually result in a live birth. And then part of the whole idea of thinking originally where it was less than 30% of conceptions actually result in a birth, and as of like I said, a week ago when I heard the doctor on the radio talking about it, now it's less than 20%, is that when they would take spontaneous abortions or even people who were having difficulty with pregnancy and they had to abort, they found that about 50% of these actually had these flaws. And these were at the point where they actually knew they were pregnant. So they were pregnant, they were looking at them and analyzing them. Like I said before, who knows how many within the first 4 weeks roughly have conceived and then lost the baby. And then hopefully this makes you feel better. 1 in every 150 live births actually has a chromosomal abnormality. So there are, 800 students here? That means 4 of us are running around here with a chromosomal abnormality? Probably me. I would bet it's me. And I mentioned this before, karyotype. What they do is they take that, what kind of cell they typically take? A white blood cell. Yep. They catch it in mitosis, they freeze that sucker, they fragment it apart so you see all the chromosomes nice and coiled up and tight. And then they arrange them from the longest to the shortest, and they lay it out like this, and it's called karyotype. So we go from the longest all the way down to the shortest for the autosomes. So the first 22 of the 23 are autosomes. What's the last one? The sex. I said before, the Y is just a little fragment. So they think that it had more purpose a long time ago, but we've evolved not to need it, except for that whole male thing. So, what would happen if the SRY gene were defective, the thing that makes us male? What would happen to it? Yes. We default to female. Yeah, so we just default to female. So there could be females running around there that actually have a Y in there that had a defective mutation. I think there was an episode of House about that, actually. How many people here watched that show? I'm curious. I think it was the one about the model. Maybe she had prostate cancer or something like that. But always interesting. And then you just have to remember the terms again. So somatic cells are your body cells, and they're called diploid because it has 46 chromosomes, which are 23 pairs. The gametes only have 20. The egg only has 23, the sperm only has 23. Why do they only need 23? Because they're going to meet up and they're going to form one somatic cell, which is the first cell of a brand new organism. And we've already gone through this several times. Okay, some of those terms that the book uses. So euploidy means that they have a normal number of 23. Multiple of a normal number. All 23 chromosomes are there. Polyploidy means that they have too many copies of the 23. So when you have haploid in the sperm, that's haploid. That's the euploid. It's a normal set of 23. If I have two of them, it's called diploid. And I have 46 chromosomes, which is a multiple of 23. But if I have three sets, is that normal? No. For us, an egg to be fertilized with a sperm should only have two sets of 23 now. So what could have happened is it got fertilized by what? Two sperm on accident. That's one way. And it's called polyploidy. Too many copies of the whole set of chromosomes. So how many would that have? 23, 23, 23 is 69. 69 chromosomes. Not compatible with life. If another accident happened and the egg accidentally had 46 chromosomes because it didn't separate properly and it's floating out there with 46 chromosomes, and let's say the sperm has 46 chromosomes that had the same fluky abnormality, and they meet up, now how many do they have? Yeah, they have 23, 23, 23, and 23. And that sucks, but that's 46 times 2, 89, 2. Oh, it's right there. Why are you so fast? So 92. And that's called tetraploidy because they have tetra means 4. Triploidy means 3. Either way, these are both poly. There's too many. Not compatible with life. And then they find that if it starts developing, then they're going to result in miscarriage. So 10% of all the miscarriages out there because this actually happens. It's a weird accident. Things happen. It could be a really fluke accident and one egg got fertilized by 3 sperm. It could cause the same thing, tetraploidy. Neither of them are compatible with life. What's interesting is that you actually have some cells in your body that have multiple copies. Your liver does. Your liver is one of those things. But it's not compatible with a stem cell. Okay. Aneuploidy. So if euploid was normal multiple of 23, what's without that multiple of 23? So that means that you don't have a full 23. One of those can be missing. 22 of them can be missing. But you can't multiply it by 23 and get an aneuploidy. It's something weird. So it might be 45 out of the 46, which is not divisible by 23. It might be 22 of the 23, not divisible by 23. So a lot of times what will happen is the ones that are compatible with life, you actually see too many of one chromosome. So if a normal life is 46 chromosomes, trisomy of just one, how many chromosomes do they have? 47. that's actually compatible with life. And we'll talk about one, which we've actually mentioned, where they get 3 sets of one chromosome on accident, and it's called Down syndrome. Or trisome Down syndrome. And then monosomies when only one's there. So how many would they have? 46? They have 45. So monosomy is not compatible with life. Trisomy is. The moral of the story is that it's better to have a little extra than a little less. How that happens is a process called disjunction fails. Normally in disjunction, you have the parent cell and it splits. And when it splits, it should split everything in half. But what happens is something called non-disjunction where it doesn't split. So this egg, if this were an egg, has nothing in it. Is that going to be incompatible with life? No. But this one has 2 sets already. So I was talking about before, if this egg met up with a sperm that looks exactly like it, we have 4 sets of chromosomes all together. Anywhere through this non-disjunction, if this thing doesn't split properly, ideally it should split so that every one of the offspring has 1 set. So 22, 22, 23, and 22. In this situation, they split so much that you got 3 in this one and only 1 in that one. Not good. It's called non-disjunction. Anybody remember the phase where the cells actually rip the chromosomes apart? So prophase, they all condense together. Metaphase, they all line up at the plate. And then anaphase, they rip apart. The anaphase, if it doesn't rip properly, it causes non-disjunction.
Just going back to that fiber, how does the one come down from nothing?
It's fertilizing. Fertilizing, yeah. Right. So it said fertilization. Okay. And then aneuploidy, so you can have partial trisomy, which means only a piece of one of the chromosomes is added. So not a whole chromosome, but just a fragment of it. Which is still pretty bad because those genes on that fragment, when they get turned on, you're making way too much of this stuff. And then sometimes you get something called mosaic. And these are interesting because sometimes an accident happens that when the cell's splitting and replicating, those stem cells, they don't replicate perfectly. You have this flaw. And they call them mosaics because half their DNA looks one way and the other half looks a different way. The term, if you ever take a genetics class, is called chimeric. And I guess in Greek, it's Greek, or I think it's Greek. The chimera is that thing that looks like a lion but has eagle wings on it, where it has DNA of a lion and an eagle, basically.
Is that compatible with life?
That is compatible with life, actually. Okay, and then Down syndrome. I don't know about the whole eagle mating with it. Yeah. It took me a second to think about that one. Maybe I should clarify. I don't know. An eagle mating with a lion sounds like a bad. Yeah. That's like a fish mating with a polar bear. There's just something wrong with it. I'm going to drop that right now. Anyway, so Down syndrome, it's an aneuploid, which means do they have a normal number of 23 or multiple of 23? Nope. It's trisomy, which means that they have 1 extra chromosome in addition to their 46. And it's number 21. So trisomy 21. This is the bulk. So the bulk of these happen because of this trisomy 21. There's actually one other form of Down syndrome we'll talk about later. It's because of a real weird mutation fluke. But when you look at the number of births, 1 in 800 live births actually happen. There are a lot of miscarriages because of this trisomy, so it's not always compatible with life. There's flaws. It's hard to deliver a child that has this flaw. When you look at the characteristics, some of the physical characteristics, so psychological, I guess, it's because of the connections in the neurons. Mental retardation, low nasal bridge, epicanthal folds. When you look at it, epicanthal folds. If you look at the Asian population, how it has more of an almond-shaped eye, the epicanthal folds are that lengthy part right here. So if their eyes are a little bit more almond-shaped, the lower nasal bridge, like I was talking about. Another one you want to jot down, I don't know if it was on the last slide, is heart defects. There are a lot of heart defects. And if it wasn't on the last slide, increased risk of leukemia too. Treating tongue, poor muscle tone. Nope, neither of them were on there. So I'm glad I said those. And then when you look at the maternal age, maternal is referring to who? Mom, right? So you can see the risks are skyrocketing after the age of 35. That egg has been sitting there for 35 years, sitting dormant. Every time she's gotten an x-ray, every time she's had exposure to cigarette smoke, toxins, alcohol, all these things can get in, go down there and cause mutation. Even just old age of the egg. Right? You want to remember those characteristics. 97% of all Down syndromes are because of this aneuploidy situation. 97% of them. Like I said, there's one other flaw that we'll talk about in just a little bit. And about 50% of them have congenital heart disease. Okay, sex chromosome ones. So, when you look at the different sex chromosomes, the first one, metafemale. So, that's double check. But metafemales is when they have 3 X's. So, for them to get 3 X's, who donated the extra X? Had to be mom, right? So, when dads split, and they go one way to another, one of those X's goes to a sperm, a Y goes to another sperm. There's no way you get 2 X's from dad in the same sperm. It's either going to be X or Y, and as long as there's a Y in there, guess what it becomes? A male. So it becomes a male. With a female, with mom, she has 2 X's. If they don't disconnect and they both go in the same one, she passes 2 X's down. So it's a form of trisomy still. The 2 X's go down, and then when it meets up with an X from the sperm, now there are 3 X's in there. They call them metafemales. And you can see the symptoms. They don't always result in this, but it can cause sterility. Irregular menstrual cycles, potentially mental retardation. And then if some accident happens, the mom gives even more X's. So if mom has 3 X's in one and dad contributes to X, it can become even worse. So symptoms get worse the more X's that they have. And I always want to throw out some kind of female joke there, and I just feel like it should be. So more X's you get really for the situation. I'll let you work on that. Anyway, and then the next one is Turner syndrome. So instead of having too many X's, they only have 1 X. So the last one had too many. This one just has 1 X. So there's no Y. If there were a Y, what would it automatically be? A boy. And then the characteristics, typically absence of ovaries. Accidents still happen sometimes. They have ovaries, but it's rare if they have the ovaries. Their stature, short. They get what they call a wet neck. So it doesn't seem like they have much neck. Usually when you see this in the physiology class, I think they use another picture where basically she looked like a wrestler. There was like no neck up there at all. Kind of broad. They have what they call a shield chest. Very broad thoracic cage. It goes flat straight down. It's webbing in the neck, shield chest. Short stature. So, and down here it says high number of aborted fetuses. So obviously some of them actually have ovaries, but they're switched along. And the most likely link is probably because of hormone issues. Next one, Klinefelter. So with Klinefelter, they have 2 X's and a Y, which means they're automatically what sex? Yep. So they have the 2 X's, but they also have that Y. So they're kind of like in between. They're male genetically, but they have a lot of feminine characteristics. They're really tall. They're usually over 6 feet tall. They have small genitalia, so more feminized. They usually get gynecomastia, which means they start developing breast-like tissue. So their male organs are smaller, but they have more feminine characteristics. Like they don't grow a lot of facial hair, have problems growing their facial hair, not a lot of chest hair. But they're really tall. So you can see XXY or even XXXY. So So mom has an aneuploidy, dad has an aneuploidy. The egg and the sperm came together, and they got two X's and two X's and an XY. All right, a lot of the men that have it don't even know they have it. So they don't make testosterone very well. They go through a growth spurt around the same age as a female. They start getting really, really tall, and not producing a lot of testosterone. So what's one problem you might have without much testosterone? Sperm production, right? Reproductive ability. So decreased reproductive ability. IQ drops with the more X's, just like we saw before. So they're why they have receptors for the testosterone, they just don't make a lot of testosterone. So is there any fix that they could do for these people? When would be a good time to start getting their testosterone? Puberty. Yeah, puberty they can start getting their testosterone. Okay, so those were some sex links. And then chromosome breakages. If a chromosome breaks, one of the things that does this is something called clastogen. A clastogen, typically like X-ray radiation, chemicals, viruses, they get in, they break the chromosome apart. Your DNA, like I said, you have repair mechanisms that see this break and they, whoa, and they come back in, they try and fix it, but it's not always perfect. So with chromosome breakages, a couple things can happen. Either that cell can die because you've got the flaw, or it can try and repair itself. And the repairs can often cause mutations. But a clastogen is something that gets in and can break it. A virus is getting in, and when it's breaking the DNA, it's actually trying to inject itself in the DNA and just breaks the DNA apart on accident. Ionizing radiation just goes through and shatters it, and we'll talk about this more with cancer. But things like cigarette smoking can do it, X-ray radiation, etc. So in this situation, you're looking at the whole chromosome. These aren't the base pairs like you saw before. But you can actually come in here and split this part up. You knock it free, and it's gone. And then when you have the repair mechanism trying to fix it, it actually shortens it up, and those genes are completely gone. It's not typically one gene, it's usually multiple genes that you lose. And one of the common examples is called cri du chat, and it means cry of the cat. And if you look at the short arm of chromosome number 5, it breaks off and they actually lose little fragments of it. But it's compatible with life. Unfortunately, it comes with microcephaly, low birth weight. When they cry, they sound like a cat meowing. That's why they call it cry of the cat. And they usually outgrow that around by the time they're 2. So they lose a whole section. We've lost genes in there. Another problem is duplication. So when your DNA is trying to replicate things, sometimes it can copy another piece to plug it back in, and they accidentally duplicate that section and plug it on in. So now you've got not a lost segment, but you've got an abnormally long segment. Duplication. And then every time you want to turn on one copy of that recipe, the other one turns on at the same time. So when you're making a product, what usually happens? You make too much of the product, yeah. And a lot of times those will actually clog up functions in your body. An inversion, so you break it on a chromosome, and when it goes to patch it together, you can't remember which way it goes, so you got a 50-50 shot and it plugs in. If it plugs it in backwards, it's called an inversion. So instead of having gene A, B, C, D, you might have A, C, B, D, where it took those middle ones, they broke out, and it reversed it. So A, B, C, D, E, F, G will become A, B, and then you can see these two got the ground. It plugs in around reverse. This position effect means that if normally I'm making a batch of cookies and I go eggs, eggs, vanilla, all the liquids, and then I go to the dry, what would happen if I took 2 of the liquids and then added the dry and then took the liquid and added the other part? You ever try doing that? There's a reason why you mix all the dry products and the wet separately and then combine them in. It makes a mess out of it, right? So it's the same thing here. You get this position effect, which means that instead of making the normal order, you actually mix these things around and screw everything up. So you take it and grab it in the wrong order. Position effect. This one, you want a little star somewhere by it. That translocation, when we talked about Down syndrome before, I said 97% of Down syndrome is because of that aneuploidy or that not ability to break apart properly, which is called non-disjunction. The other 3% are because of translocation. And it's this one right here. Robertsonian translocation. Another eponym. The doctor discovered what this was and labeled it. So 3% is because when the gene or the part of the chromosome breaks, when it puts it back into the chromosome, it actually puts it in a totally wrong area of the chromosome. And the Down syndrome, what happens is, you remember the name of the chromosome that was associated with the other 97% of the Down syndrome? Number 21. When that piece of 21 breaks off, instead of just having a separate chromosome, what will happen is it takes that chromosome and sticks it onto the chromosome number 4. So you have this really weird structure on number 4. So it's not technically a trisome where you have 3 chromosomes. You have an extra long number 4. It's called Robertsonian. And I said about 3% of all Down syndromes are because of that. And then why you want to remember it is because if two parents have Robertsonian, they're more likely the recurrence is higher to have another two Downs, and another, and another, and another. So it increases the risk of having multiple Downs children. And then the fragile sites. So fragile sites are exactly that. When I talk about mutation hotspots or weak spots, that's what's going on. These are fragile. What's the fragile disorder that you know I'm going to talk about? Fragile X. Fragile X is a lot like Huntington's. Remember how I said in Huntington's you have that repeated pattern over and over and over again? With Fragile X, you do exactly the same thing. You get these breaks or gaps, and with repairing it, it just makes that gap or that break longer and longer and longer. So as I'm patching, I make this long strand of worthless crap DNA. And there it is, Fragile X. Take a guess at what chromosome it's on. The X chromosome. Right. So at the end of the X chromosome, it gets longer and longer and longer and longer. And it's the second leading cause of mental retardation next to Downs. And then you know what happens. Since the girls can have one normal one and one bad one, then they get the normal one that takes care of it if that X breaks or fails. But the men, if that X breaks, then they get problems. So it happens more in males than females. Could it happen in females? It could, yeah. They'd have to get 2 bad genes. And it's the same thing like I said before. They get that CCG codon over and over and over. It's like the stutter or the skip. Dog, 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 dog. The longer that is, the weaker it gets. The worse the symptoms. And in some ways that they can fix this, gene therapy. So with gene therapy, the tricky thing is that we're going to manipulate viruses. These are viruses. They mutate easily. So is gene therapy always a good idea? You should watch, what's that movie with I Am Legend with Will Smith. So the basis of that movie is that they were doing gene therapy and they were working with a virus and the virus mutated and then caused a big problem. But what they do is they take a virus and they ideally take a virus that's not super bad or dangerous. And then they put the good gene in it, because what do viruses want to do, you know? They want to go into your DNA and inject themselves in your DNA and patch themselves in. So if they take a good gene that they want to get into somebody and stick it into this virus's DNA, then hopefully the virus gets in. Where would they have more effect? Would they have more effect in that DNA, or would they have more effect in an RNA virus? If they were to manipulate the RNA virus, it would actually be more powerful. Right now they're looking at DNA. So what do you know about DNA when it gets in the cytoplasm? Yeah, so there's a high likelihood it's going to get destroyed. DNA viruses are still dangerous, but not nearly as dangerous as RNA. So one of the problems is that you can inject someone. If you want to fix their gene, you can inject them with this virus. But what do you know about viruses in your body? What's your body want to do? Kill it. So you have to hope you infect enough of their cells fast enough that it actually makes a difference. So there are a lot of flaws with using viruses for gene therapy. But typically what they'll do is they'll inject this good gene into a DNA or into a virus and then inject it into you and hopefully get to all of your cells. But less than perfect. for it to be more ideal, they'd have to keep changing the virus. They'd have to inject the same person with lots of different viruses with all that same gene in it, because after you've been exposed to one virus, guess what your body does? It builds immunity to that virus. So now that virus is worthless. You can inject them again, and it does nothing at all because their immune system killed it right away. So you have to change it, and change it, and change it, and change it. So it's a lot of good idea in theory, but it's not all that applicable. But cystic fibrosis is one of those things they really want to try and change. If they can inject the right genes in to make the little change, they're wrong. What do you think they'd have better success with? Single gene or a polymodal? Single gene, because they know exactly where to target. Polymodal a lot tougher. And then environmental factors. So some of the flaws, you probably know this. Smoking associated with low birth weight could cause problems through alcohol syndrome. So there are a lot of environmental things that can change the genes too. And then if it's a congenital malformation, is that okay or bad? It's bad. Yeah. So sometimes you just get those anomalies, like an extra finger. That's okay, but sometimes things like drinking gets across the placenta, gets into the baby's brain, slows down the neural connection. And the brain doesn't know how to develop properly at that point. So they'll always have symptoms for the rest of their life and fetal alcohol syndrome. The big scary period, day 15 to day 60. So if you expose it to toxins between 15 and 60, that's when the organs are actually growing. So they call it organogenesis, which I think is on the next slide. Yeah. So you can see most of the organs are developing from week 2 all the way up to roughly week 8. So if you get high exposure to alcohol through here, it's most likely going to be the heart, the extremities, or the eyes that are affected. If you have alcohol exposure within the first few weeks, so within 2 weeks, does mom always know she's pregnant within 2 weeks? No. My wife did this because we knew we kind of wanted to, so we went to the Dollar General and bought like 50 pregnancy tests. So when we would go out, anytime we would go out and have drinks, we'd show us 50 pregnancy tests, just in case. So keeping our fingers crossed. It's a good idea. Right? Unless you're not planning on it, then you don't think that far ahead. This next word, teratogens. Teratogens are chemicals that can get in and actually affect organogenesis. Back in the, I think it was the late 50s, early 60s at the latest, Great Britain actually came up with a brilliant chemical that prevented morning sickness. So it was called thalidomide, and the parents would take, or the mother would take it, and she wouldn't have morning sickness. So she felt great during that time, but unfortunately, she took it at a time of organogenesis for the extremities. So during this phase, when she was taking it around morning sickness, she was affecting the arms and legs, and they were actually born with what they refer to as flipper limbs, because they didn't have hands or legs that were developed, and just had basically structures coming out of their arms, if they even had that. But it's called a teratogen. So chemicals that affect organogenesis are called teratogens. And then things like radiation. Like I said before, they used to actually adapt thermometers with X-rays to see how far they were along and what was going on. Okay, it's 3:43. No time to start cancer. So you should be able to take the lecture quiz over cancer tonight.
Is there a phone?
There should... Oh, that reminds me. Thanks. This thing... I'm going to do the front side as homework and the back side as extra credit. So the front side worth 10 points, the back side worth 5. The video is... I think there was a link online, right? You saw it? Okay, there's a link online. If you can't open it up, this is what you want to Google. If something happens, because sometimes when a lot of people watch the video, since it's actually copyrighted, they will block it. So just go to Google and I can't remember what the key word. It's yeah. I used to know this. Oh, yeah, and don't leave without turning your last homework in. Just drop it on the front corner on your way out so I can get it.
I wrote some stuff to clear most of
Perfect. This is what you Google. It's called Google autopsy, life, death, cancer. Just those 4 words. And when you click on video, so there's step number 1. Number 2, you just click on the top of Google. You get like web, images, places, news, whatever. Does everybody know what I'm talking about? On the top of Google. Go to video. And then you'll click on the one, it's like 45 minutes long. It's called Autopsy, Life and Death, Cancer. And right when you open it up, it's that guy, Gunther von Hagen. You don't have to write this down because I think it's actually on the handout. But anybody know who he is? Remember the Body World Fascination? He made the Body World. So what he does is he goes through and does. There are 4 videos on life and death. One is, and I'll give you extra credit on another one, it's actually a heartbeat, and he makes a sever in the femoral artery, and they count how fast it would take to lose 2-3 months of blood, and how long it would take to die. Yeah, that's it. But we'll do those. But this one, you're looking for the one with Gunther von Hagen, and like within the first couple minutes, you can tell it's him. He has a funny hat. And he speaks with a very strong German accent. But all these questions are right in order. All you have to do is watch the video. It's like 42 to 45 minutes.
You saw my
Your handout?
Thank you.
And yeah, they're all right in order as you go through it. So if you accidentally see the next one and you missed a question, just rewind a little bit and you'll find it. Okay, we skipped the second break, so we're getting out 13 minutes early.